All right, so this week we're going to be going over genetics, and here are the basics. So let's get started. Remember, if you click on the microscope, you can um, see the websites. So just keep that in mind as we go through the notebook. If you want to learn more outside of the notebook, please just click on the microscope and see what's up. So for slide 2, you're going to learn the stages of mitosis. So the stages of mitosis, here's a good old meme for you. When your sister steps on your toe, you go, ouch, mitosis. So mitosis is how body cells are produced. Body cells are anything that are not sex cells. When a cell goes through mitosis, two sister cells are made. So you'll have one cell, the DNA and the chromosomes will double, and then those cells will divide. That one cell will divide into two. So now you will have two cells. So you go from having one body cell to having two body cells. Here are the phases or the stages of mitosis. There is interphase. That's just the beginning of division. The next we have is prophase, and you'll start to see that there will be these spindles forming, and you'll start to see that your chromosomes are starting to condense, and you'll start to see them. My metaphase, the chromosomes line up in the middle, M for meta, M for middle. Next, we have anaphase. Anaphase, they start pulling away. So A for ana, A for away. So the chromosomes start to pull away at this point. Next, we have telophase. Telophase is toward the end. So telophase is toward the end, and you could see that the two nuclei are starting to form. So that's what these purple things are. That is the nuclei. The two nuclei are starting to form. Then you have cytokinesis. Cytokinesis is when the cell starts to form two separate cytoplasms. So cytokinesis, two cytoplasms. Okay? So just you will see all different variations of this idea. The main thing that you need to know is that the chromosomes start to appear. They line up in the middle, they pull away, and then the two nuclei form, and that's when you start to get your two cells that will form. So they will separate and they'll pinch off into two separate cells. The next one we have is slide 3. Slide 3 is where all of these were over here on the side. If you can't see them easily, just click and drag them to the center and then figure out where they go from there. So, interphase is the phase that the cell spends most of its time. That's where the cell grows and develops. Prophase, the chromosomes are visible. Metaphase, the chromosomes line up in the middle of the cell. Anaphase, the chromosomes divide into two sets. And in telophase, cells begin to separate. And in cytokinesis, that is when the cytoplasm divides. So cytokinesis, cytoplasm. PMAT, so prophase, metaphase, anaphase, telophase, those are where the cell is actively dividing. Interphase is where it spends most of its life. Cytokinesis is when the cytoplasm is dividing. So if you want to remember PMAT for the order for the ones in the middle, go for it. If you want to remember IPMAT C, I don't see why that would be an issue. If you want to come up with a mnemonic for it, please do so. However you want to remember the stages of it, go for it. So interphase, cell develops and grows. Prophase, chromosomes are visible. Metaphase, chromosomes line up at the middle. Anaphase, chromosomes start to divide into two. So I always remember anaphase pulls away, the chromosomes are dividing. Telophase, cells begin to separate. And cytokinesis is when your cytoplasm divides. Slide 4, you are learning a little bit about meiosis. Meiosis is how sex cells form. I would recommend that you watch these videos down here to understand meiosis a little bit more. I'm just going to go over it very briefly here. So, meiosis halves how many chromosomes you have in your body cells. So, eggs have 23 chromosomes. Sperm have 23 chromosomes. When the egg and the sperm meet, they form a full cell, which is 46 total chromosomes. So there are 23 chromosomes in an egg. So there's half of the amount that you need for your body. There are 23 in the sperm. And when the egg and the sperm come together, they make a typical human body cell, which has 46 chromosomes total. Okay, so that's all you really need to know about meiosis. Like I said, I do recommend you going and watching this. Meiosis divides. There are 2 stages of meiosis. By the end of meiosis, there are 4 total sex cells. Okay? There are 4 total sex cells by the end of meiosis. They are also called sex cells or gametes. So there are gametes by the end. And each of these cells have 23 chromosomes. On slide 5, you're learning the differences from meiosis and mitosis and what both are. So, meiosis, you make haploid cells. So, haploid means half. So, half of what you normally have. There are 4 genetically different cells by the end. So, the 4 cells that are made in meiosis are all different. And they make sex cells. Remember, sex cells also, another word you can use for that is gametes. Mitosis, at the end of mitosis, you get 2 identical cells. You get 2 identical sister cells. The body cells are called somatic cells. You need to know that. Somatic just means body cells. Diploid, mitosis makes diploid cells. That just means that they have the amount of chromosomes that you need. So our cells are diploid. I put this here. It technically should be in both, but I put it in mitosis because it makes more sense to me. Because um in meiosis, there are multiple stages of these, so I put it in mitosis because this is really what makes sense for mitosis. There are multiple stages in meiosis, but they're not quite the same. And both here they are both a type of cell division. Slide 6 is just the differences between chromosomes, genes, and DNA. So, chromosomes are a thread like structure of nucleic acids and proteins found in the nucleus of most living cells. Let me change this text color. So, okay, chromosomes are thread like structure of nucleic acids. So, chromosomes, I will show you the picture that is on the website. It makes a lot more sense if you do. So, in the cell, you have chromosomes. Chromosomes are made up of DNA. And then on that DNA are genes. So, what are genes? Genes are just a unit of heredity which is transformed from a parent to an offspring and determines some characteristics of the offspring. So, genes are just a unit of heredity. It is just a set of DNA that codes for something. So, your eyes have genes, your skin has genes that code for it. Um, Everything that is on your body has genes that code for it. If you're lactose intolerant, that's a gene that is coded in there. So things like that. And then what is DNA? DNA is short for deoxyribose nucleic acid. If you want to say that out loud so you sound, fancy, go for it. Makes you feel smart when you can pronounce it correctly. So it's just deoxyribose nucleic acid. That's what DNA stands for. Early studies of DNA just suggested that DNA was very simple. However, in the 1940s, they discovered that genes of bacteria are made of DNA. So we started recognizing that DNA was very simple. We thought it was very simple, I should say. And then we started recognizing that even the most simple organisms that we could think of had DNA. So, what was going on with that? Why was DNA the thing that tied everything together? So, we're going to, in a couple slides, we're going to learn more about the scientists that did research on DNA. We're also going to learn a bit about the structure of DNA. So for slide 7, you were to click and drag the letters to where they are paired correctly for the structure of DNA. So adenine and thymine pair. So A and T pair with each other. Cytosine and guanine pair. So cytosine and guanine pair. I went ahead and I made the orange ones here T and the gray ones C. The peachy ones yellow and the blue ones green. So just the blue ones green. The blue ones with G, sorry. Just so that we could start to see that it does matter what we label the DNA. The T and the T have to be the same, look the same. The A and the A have to look the same. C and a C and the G and a G. Okay? So T and A always pair. G and C always pair. I always remember this because the G and the C both kind of curve, and that's just how I remember it. So like the curved letters go together. There is a picture for you here of the phosphate sugar nitrogen backbone. So all of these base pairs are broken down like this. So the phosphate group is over here by itself. There is a sugar. Remember when we talked about glucose, I said that a lot of words that end in OSE are sugars. Well, think about DNA. DNA stands for deoxyribose. And guess what's in a nucleotide? Sugar. So you have the ose in there. Ose is sugar. So there is a sugar and then there is a nitrogen base. So the nitrogen base is actually what connects to each other. The sugar and the phosphate make up the backbone. It's called a sugar phosphate group, a sugar phosphate backbone. So that's the backbone of DNA. The nitrogenous base is the base pairs that pair with each other. For slides 8, 9, 10, and 11, we're going to go over scientists. I'm going to go ahead and start with Aaron Charkoff. Charkoff discovered that there are differences in the DNA of different species. Charkoff's rule states that adenine will only pair with thymine and guanine will only pair with cytosine. So Charkoff figured out a way to see the ratio of adenine and thymine and guanine and cytosine. And he noticed that there were the same amount of adenines as there were thymines, and there were the same amount of guanines as there was cytosine. So that's how he came up with this rule. So adenine only pairs with thymine, and guanine only pairs with cytosine. So A with T, G with C. For slide 9, we're learning about Rosalind Franklin. Rosalind Franklin, let's click on the microscope. Let's click through this and go to Rosalind. And we'll see that Franklin worked at the King's College and used X-ray crystallography to obtain pictures of DNA fibers. Her results were then unknowingly shared with Watson and Crick by her boss, Wilkins. Her data was instrumental to Watson and Crick's discovery of the structure of DNA, yet she was barely given credit and has not received a Nobel Peace Prize for her efforts. So Rosalind Franklin took these pictures of these cells, and she just kept taking these pictures and kept taking these pictures using X-rays. And she was able to get pictures of DNA, and her findings were instrumental in the discovery of the structure of DNA. So she found this X. I think it was called Photo 51, and it's an X shape. And she couldn't quite figure out what was going on, why she kept seeing the X shapes. And her boss at the lab took her data and shared it, her pictures that she took, and shared it with Watson and Crick. So Wilkins, her boss, shared it with Watson and Crick, and this is without her knowledge. And Watson and Crick used the picture to help discover the structure of DNA. And Watson and Crick received a Nobel Prize for their efforts, and Rosalind Franklin did not receive any credit for her help toward the identifying the structure. Slide 10, you're answering about Watson and Crick, and if you click on the microscope, you can read a little bit more about them. They did win the Nobel Peace Prize. Peace Prize. Wow, sounds like I need lunch. For their medicine in 1962 for their discovery of the structure of DNA, Watson and Crick used Franklin's X-ray to figure out what was going on. And with that X shape, what they discovered was that what it actually was taking a picture of it from above. So if you take a picture of DNA from above, it looks like an X because of that double helix shape. So Watson and Crick were able to figure out that it was a double helix. That is the shape of DNA, a double helix. You see how these strands curve like this, and there's two of them? So it is a double helix, and they twist. So if I were to take this DNA strand and I twisted it up, I could get it really compacted without there being any issues with that DNA. By issues, I mean some kind of mutation, some kind of damage, something like that. It's able to twist down and condense because of that shape. Describe the shape of DNA in your own words. So DNA has two strands that twist around each other. Think about it as like a spiral ladder. So it kind of looks like a ladder, like a staircase, like one of those fancy spiral staircases. Kind of looks like that. Slide 11, we're learning about the father of genetics. That is the name that is given to Gregor Mendel. Why is it? Okay, there we go. So, he was a botanist who started to lay the foundation of genetics. After Mendel completed his studies, he moved to a monastery and became a monk. While he had access to the garden and began experimenting with the pea plants. He observed different characteristics of the plants and would crossbreed them to try to get different traits. Through his experiments, Mendel coined the terms recessive and dominant, referring to the different traits. So, Mendel studied pea plants. Let's say he would take a yellow pea plant and he would cross it with a green pea plant, and he would determine which trait was dominant by that. And that is where we get Punnett squares from. We will learn a bit about Punnett squares in the next activities that we do. But right now, there are no Punnett squares on this notebook. But there will be, so don't worry. I love Punnett squares. I think that they're super fun. Um, but that's just me. I like puzzles. They're like big puzzles. They're great. What kind of plant did Mendel use? Mendel used his pea plant. He used a pea plant. And the terms that he used to describe these traits we still use today. There are dominant traits and there are recessive traits. So dominant traits means that it covers up the recessive trait, and recessive means that you need two of those genes in order for it to show. So there are lots of dominant and recessive traits, and we'll talk more about those here in a minute. Slide 12, you're learning about inherited versus acquired traits. So if you click on the microscope, it'll take you to this page. At the bottom of that page, it tells you the difference between acquired and inherited traits. Acquired traits are things that you need to learn. So these are things like talking, a dog going and fetching a ball, being able to play basketball, soccer, any sport, being able to play a sport. That's an acquired trait. Inherited traits are things like your hair color, your natural hair color, and your eye color, skin color, whether or not you have freckles, how long your fingers are, things like that. So inherited traits are your eye colors, uh, hitchhiker thumb. That's if you can like bend your thumb back, and freckles. So freckles are, if you are out in the sun, you might get more freckles, but you're still inherited the trait to get freckles. So there's that. Acquired strong muscles. Everyone of course has a different muscle mass naturally, and they genetically inherit that. However, everyone can train to get stronger muscles. Everyone can acquire stronger muscles. Swimming, you got to learn how to swim. You don't just know. And it's a acquired trait that you need to learn. And playing the guitar, playing any instrument is an acquired trait. Some people may inherently understand it easier and be able to understand music theory better and might have the ear for it better, but still the act of learning how to play it isn't inherited from your parents. It comes from practice or the environment that is around you. It doesn't come from your genes. So inherited comes from your genes, acquired comes from your surroundings. Slide 13, we're talking about traits. We're talking about alleles, genes, genotype, phenotype. So all those words, you're like, what in the world are you talking about, Miss Foley? Well, let's talk about it. So an allele is just a different form of the same gene. So you can have two different alleles for a trait. So you'll get one allele from one parent and one allele from another parent, and those make your genotype. Okay? So you have two sets of genes for every trait. So two sets of genes for every trait. So not two sets. You have one set, and a set is 1 and 2. So you have two genes for every allele. Okay? So you have one allele and another allele. So one comes from one parent, one comes from the other. Okay? So different form of the same gene. That is all an allele is. An allele is just a different form of the same gene. What are different alleles for eye color? So here are some alleles that you may have for eye color. There are blue, brown, green, hazel, gray, any color of eye that is naturally occurring. All options of eye color they have alleles for. There are alleles for those types, those genes. Blue, brown, green, hazel, gray. So many different eye colors, right? So all options of eye color. Now we're going to learn about the difference between genotype and phenotype. I haven't said this already, but I'm going to say it for this slide. Watch these videos. They're 1.5 minutes. Watch them. They will help you out a lot if I am confusing you. Genotype is the pair of alleles. So this is your genetic makeup. Your genotype is your genetic makeup. So these are things like capital B, capital B, lowercase b, b, b. So each of these letters stands for one gene, one allele. Okay? So the capital B, let's say you got the capital B from your mom, you got the other capital B from your dad. If this was your genotype. If this was your genotype, then let's say you got the capital B from your mom, you got the lowercase b from your dad. Okay? And then for the little b here, the lowercase b, then you got the little b from your mom, you got the little b from your dad. Okay? So just keep that in mind when we're going to go over Punnett squares, and when we do, this will make a lot more sense. So I always remember this as G for genes, G for genotype. Okay? The next thing we have is phenotype. Phenotype. This is the physical appearance. So let's say that this capital B was for brown hair, and this lowercase b was blonde hair. So if there is a capital letter, then your hair is going to be brown. Okay? In this case, genes are not that simple, but we're simplifying it so you can understand. So you got the capital B from your mom, the capital B from your dad. So chances are, in this situation, both of your parents they have brown hair. Now, let's say that you got capital B here from your mom and lowercase b here from your dad. So that means that more than likely your mom has got brown hair and your dad has blonde hair. Naturally occurring blonde hair. Okay? So let's think of it that way. And you're going to have brown hair because you have a capital B and you have a lowercase b. Now, if we're looking at the lowercase b, chances are your mom has blonde hair and your dad has blonde hair. And so you're going to have blonde hair. It's not that simple. We'll learn more about it in Punnett squares, but just giving you a heads up is genotype is the actual genes and phenotype is the physical appearance. So I always remember this as the pH, so phenotype, and the pH for physical. So pH for phenotype, pH for physical. Here you are clicking and dragging these boxes to where they go. So dominant traits, like I was saying, if it has a capital B in it, then it is going to be a dominant phenotype. So this has a capital B, this has a capital B. This has a capital B, this has a lowercase b. So even though it has a lowercase b, it still goes over here with the dominant traits. Over here, we have the recessive trait. The only way a recessive trait shows up is if you have both of the genes from your mom and from your dad in order to get that recessive trait. Okay? So your mom has to give you a recessive trait and your dad has to give you a recessive trait in order for you to have the phenotype for a recessive trait. And that'll make more sense too when we do Punnett squares, but for now, just keep it in the back of your head. For slide 14, you are just doing this by yourself and you're going to summarize, try to make it a complete sentence. So you can either read about if we should bring back animals from extinction, or you can watch this Bill Nye video about cloning. So should we bring animals back or cloning? So summarize the videos there. And then here's a video about are GMOs bad? And another video about designer babies. So you can watch these, just choose whichever one you want and write about it yourselves. I'm not going to help you. You can do this one by yourselves.